We're live. Good morning, everyone. Everybody say hello.
Hello.
Oh, well. So we've been talking about the cytoskeleton. Specifically going through some of the components that make up the cytoskeleton. And we start out with, the word that came to mind was lame, but that's not fair. Intermediate filaments, though, they're lame. Intermediate filaments are a broad class of cytoskeletal elements, and they don't have, I don't think, a tremendous amount of homology. It's more the molecules that can form these different kinds of fibers. And so there are broad classes of intermediate filament-like proteins and things that are specific to neurons, intermediate filaments that are pretty much ubiquitous. And their main job is to provide structural integrity to the cell. Things like being able to pull apart your skin don't happen because intermediate filaments. And there are mutations that will disrupt some of the specific intermediate filaments, things like keratin, such that they don't hold together very well, and you end up with epidermolysis bullosa, I think it's called. It's a blistering disease where your epithelia starts to peel up because it's not being cemented down. And we'll see, we start talking about the extracellular matrix as well, that there are structures that are connections between intermediate filament spots between two cells, and then there are structures that are intermediate filaments between a cell and the basement membrane. And so what they're doing is allowing for mechanical forces to be transmitted across an epithelial sheet. And rather than having some kind of, just a cell adhesion molecule, which is a pretty cool thing, and they exist, you now have a cell adhesion molecule that's backed up by this fibrous material in the cell. And so it provides a fair amount of critical strength to the cell and to an epithelial sheet. That said, one of the key points that we'll see here is there are no motor proteins. There are no proteins that are able to move up and down an intermediate filament type structure. Additionally, intermediate filaments don't have a polarity. The way they assemble themselves, one end is the same as the other end. And we're going to see here as we start getting into microtubules and microfilaments, that's actually a critical point in the way these guys work. So we then turn our attention to the microtubules that are made up of the protein complex known as tubulin. There is an alpha subunit and beta subunit. There's even now like a gamma subunit, which is a part of tubulin that starts out at the microtubule organizing center. It's kind of the base. And tubulin has been seen to assemble and disassemble very quickly. It's a term that is referred to as dynamic instability. Because as people have been studying microtubules, within minutes you can have a microtubule structure form, and then just as fast you can have it disassemble. And so you've got a pool of tubulin monomers floating about in the cytoplasm, and at the given word, they can assemble up. And so a classic place to study microtubules has been in the spindle fibers. And you recall this from genetics, talking about cell division. As the chromosomes lined up on the metaphase plate, the spindle fibers come out from these specific spots on either side of a cell that are known as the centromeres, and they attach, or the centrosome, and they attach at the centromere on the chromosome. All right, so this is a structure that then reaches out and actually binds to the chromosome. And not to belabor the point, but it turns out that there are specific spots on the chromosomes that are the binding points for spindle fibers. And not surprisingly, there are no active genes in that spot. And in fact, the DNA sequences are filled with a lot of repeat structures. And it's probably a way of allowing protein complexes to form there that allow for the spindle fibers to actually firmly attach, because they're having to pull this chromosome back through something that is the consistency of jelly. And so you got to have some actually fairly serious connection going on here. So a lot of work has been done looking at spindle fibers and how they can move back and forth. Another classic model system for looking at the microtubules, I was thinking about this as a preparing my coffee, are things like flagella. And flagellated little organisms, microorganisms, might have a cell body, and then there'll be a tail that will whip back and forth. I mean, sperm have this, but lots of protists have this. There are not a whole lot of flagellated cells within your body other than the gametes in the male. There's cilia, and cilia don't necessarily have to have microtubules. Sometimes, depending on how big they are, they might. Regardless, there's a whole study about how flagella work, and there's actually a couple of figures in the book, and it's quite an exciting thing. I just go, really? We have other things to talk about. But the main point is they make use of these motor proteins. And so there are a set of motor proteins on or associated with tubulin. Specifically, they are ATP dependent, and the term is a motor protein. They are able to have a binding and hydrolysis cycle that causes them to change shape, and they can, you can see my feet here, but they can march down the microtubule. And we've got two primary motors associated with tubulin, and they're referred to as being the plus end directed motor protein called kinesin, and the minus end directed protein known as dynein. Dynein and kinesin are the two primary motor proteins that are associated with microtubules. And these guys are doing a lot of work in that there are vesicles. In fact, if you recall, we were talking about the Golgi, and at the trans side of the Golgi was a structure known as the trans-Golgi network. And it is a whole set of microtubules that lead towards the cell surface. And so you've got the dynein or kinesin attached to a vesicle, and it's actually crawling along this microtubule structure. And we can specifically think about this structure and some of the mechanics here when we think about the spindle fibers. So the spindle fibers start out at the centrosome, at this microtubule organizing center, and they're heading out. And when they hit the chromosome, they attach, and then they're going to pull back. So remember, the dynamic end is the plus end. That's the end that is being assembled and disassembled. And so what we know then is that this is the plus end here. And as it turns out, because people actually have done a fair amount of looking around at this, when you observe, and you can do some of these experiments by actually incorporating labels into some of the microtubule subunits and watching what those labels are doing as they move back and forth. And we have a model that looks a little bit like this, in which, we'll draw that one over there. So we've got a chromosome, and it goes out way out there. A lot of proteins right here bound to the middle part, the centromere of the chromosome. And the microtubule spindle fiber is coming out and attaching. Now, in order for the microtubule to actually have its motive force, we have to have essentially little feet sticking here that are walking down. They have little bows that are walking down the chromosome. They're stepping down this way. And what we know is that the spindle fiber is disassembling. So we have microtubule monomers coming off this end. And the, let's see, so this is going to be a minus end directed motor. That is the kinesin. let's see, hold on. The way I keep up with it is the plus and minus, and then in alphabetical order, dynein and kinesin. So the minus and directed is a kinesin, is going to be pulling this thing down. So the kinesin is essentially stepping ahead of the disassembly. And some of the things you can observe is that the end of the microtubule spindle fiber doesn't remain intact. And so you don't have the spindle fiber set, the little Twizzler there, and the chromosome sliding around it. Yo.
What is the minus and plus undirected protein called?
And if I messed it up, we can record this and I don't have my book on me. I'm saying it is this. We have kinesin and dynein, and specifically which one's minus and plus directed is not all that critical, but dynein is the minus in directed and kinesin is the plus in directed, and so I just screwed that up, didn't I? Thank you. We can edit that out, I'm sure. This is the dynein motor heading that way, because the plus end is going to be this side. The minus end is going to be over here. And so lots of different microscopic studies and analyses have looked at this disassembly and assembly. But here's a couple of cool facts and cool points. Because the spindle fiber has to dissociate as the chromosome is being pulled, I think of it a little bit like the cartoon where the bridge is on fire and you're running right ahead of it and the bridge is burning right behind you. There are actually some interesting drugs and things that are used in this system. Yo, question.
What was the thing that you said was walking again? Was walking down?
These are the motor proteins
Okay,
that are attached to the chromosome.
Awesome, thank you.
Also known as dyneins.
Yeah, it is the dynein motor proteins. Pretty much, I don't do microtubule studies. Dynein and kinesins are the primary motor proteins. We don't really hear about too many other ones. And so we have vesicles that are attached to them, and they're tracking up and down. And so this is one model system that's been looked at. But when we also talk about vesicle transport, we're talking about microtubule motors that are moving up and down attached to the vesicle. Anyway, the ability to monkey around with microtubules actually becomes a critical cell division interrupting system. And essentially, these are drugs that are used as cancer chemotherapies. And there's a few of them you may have heard, or perhaps. And so by blocking spindle fiber either disassembly or assembly, you can begin to affect cell division. And one of the principles behind chemotherapies with cancers is that it's based on the fact that a cancer cell has got a higher metabolic rate and is dividing a lot faster than the rest of the cells. One of the sad points about cancer chemotherapies is that they are going to be metabolic toxins, and the hope is going to be, or very often they are, the hope will be that the more rapidly dividing cells take the toxin up faster than the rest of the body does. And so, not surprisingly, cancer chemotherapies have a number of fairly ugly side effects because it's a race to see who can get knocked back first. Anyway, there are a couple of cancer chemotherapeutics that target microtubules. Specifically, there are a class of plant alkaloids from the periwinkle known as vincristine and vinblastine. And these are inhibiting the assembly of spindle fibers. They're binding to the microtubule, the tubulin monomers, and what they're doing is blocking spindle fiber formation. And if the spindle fibers can't form up, then you don't have the ability to pull the cells apart, and the cells are going to be in mitotic arrest. There are a couple of other drugs that do this as well, another one called cytochalasin. Sometimes in tissue culture or in various experiments, you want to have everything blocked in what is referred to as mitotic arrest. And you do that by incorporating these drugs that will cause the spindle fibers to not form, and consequently, as the cells go into the cell cycle, they'll go into metaphase, but without the spindle fibers, they can't pull, and everybody stops at that point. You may have seen chromosome spreads from like humans when you're doing, it is actually becoming sort of a somewhat old-fashioned technique, but you can take animal cells and you then treat them, and this is done sometimes with amniotic fluid, to look for trisomy. And there are other ways of doing it now, but this is one of the ways that it used to be done. And you would take the cells, put them into a little bit of a culture media with some antibiotics so the bacteria don't grow, and then hit them with something like cytochalasin, which is one of these metabolic inhibitors or spindle fiber inhibitors, and you cause everything to go into mitotic arrest. You then take the cells, and back in the day, you physically would stand up here and have a slide down here and drop the cells from quite the distance, and they splat onto the cell or onto the glass slide. And then you can stain, and you maybe have seen photographs of the condensed chromosomes. And essentially what you do is you take a photograph of them, and then with a little tiny pair of scissors, you physically cut the chromosomes out and you lay them out on a chart. And a trained cytologist can recognize the individual chromosomes, line them up, and make sure that you can tell the sex because you have an X and a Y or two X's, and you can look to see that you have the appropriate number of chromosomes. And this is one of the ways that chromosome analysis used to be done. I've never done it, but I've heard stories of people standing on a stool and dropping the slide, the drop, and trying to hit your little cover slip. They now make something called a cytospin, where it's got a cover slip at the bottom of a little thing that you put in a microcentrifuge, and you spin it down onto the slide. Regardless, by placing cells in mitotic arrest with these kinds of drugs, you can cause the cells to begin mitosis, but then halt. So vincristine and vinblastine are really good chemotherapeutics for different kinds of leukemias. Yes, a question from the audience.
What's the connection that was being made between flagella and the motor proteins?
The jello and the motor proteins?
The flagella.
Excellent question. The point being that the cytoplasm is really dense. And we don't really think about it like this, but it's got a protein concentration higher than what you can generate in a microcentrifuge tube. And so the viscosity and the consistency of the cytoplasm is pretty gooey. And so they have to have a fairly firm connection as they're being pulled back. That was the connection there. Is that what you were asking?
So the flagella...
Oh, wait. I thought you were saying jello. Flagella. Masks? What? I'm going to turn the damn video off. Shit. All right. The flagella was just another model system for studying the formation of microtubule structures. And I mentioned it only because there's a huge amount of study with flagella, and we'll probably look at that picture, but I find it tedious and boring, and I've never in a million years really gone in deep dive into how flagella actually operate. So if you're interested in flagella, look it up on Wikipedia. Don't talk to me about it. Actually, there's all kinds of weird structures and spokes, and it primarily works because you have one side of the microtubule structure is anchored, and then another side has got the ability to move, and you have dynein and kinesin motors that are doing that and causing the whole thing to kind of slide back and forth, which causes it to move back and forth like that. So this could become the next thumbnail for the video. If you listen to the video on Monday, I was trying to develop a thumbnail that would be different than me doing this. And the thumbnail on that video was a lovely picture of a receptor and its ligand. And I'm trying to come up with a better kind of thumbnail. It's been a long semester. Now, so the connection between jello and flagella and microtubules, not a clue. This stuff just comes out of me, man. I'm sorry. No! Vincristine, vinblastine, really good. There are a range of leukemias that are absolutely treatable because vincristine and vinblastine are so effective. There is another cancer chemotherapeutic that is also targeted into the microtubule system. And this is another plant alkaloid, actually isolated from a fungus that grows on a yew tree. It is a compound that perhaps you've heard of known as Taxol. Taxol is used a lot in breast cancer treatment. Taxol takes the opposite approach in that instead of blocking spindle fiber production, what it's going to do is it is going to block microtubule MT, for those of us that are hip, disassembly. 1S, 2S, whatever. It blocks them from coming apart. And so I've actually used Taxol for various experiments in fly embryos. You can shoot a little bit of Taxol into the fly embryo prep as you're fixing them. And when you stain them for microtubules using an antibody for microtubules, man, you have these beautiful spindle fibers just all set around. It's quite the cool thing. So Taxol is doing the opposite of vincristine and vinblastine. Vincristine and vinblastine are inhibiting the polymerization. Taxol is inhibiting the deconstruction. And because you have to have this end falling apart before the chromosome can be pulled back, or as the chromosome is being pulled back, it also will block mitosis. There you go. And let's look at a couple of figures just because we should talk a little bit about flagella.
Is Taxol related to paclitaxel, or are those two different?
It is one of those things that, like, what the hell did you say? Is Taxol related to?
Paclitaxel, I think it's called, like another common chemotherapy.
Ooh, I don't know.
Their names are similar. So
Yeah. I don't know. There's also tamoxifen, which is, no, shit. I'm waiting off on this stuff I don't know nothing about. I had to go to Wikipedia for this. So, and what's really embarrassing is this is on record, because normally I say crap that is wrong, and it just passes out, and I can maybe reflect on it at night going, oh shit, did I screw that up? And I can come in and say, okay, I messed that up. But here, it is on permanent record. That's why at the end of the semester, I'm deleting my entire YouTube channel. But we have a new subscriber. Welcome. I don't know, some dude, and it's like, what the hell, man? Yeah. Yeah, I'm glad. I hope I'm entertaining. I don't know. Okay. No, it becomes quite meta in that, there are people that are here regularly. There are people that are doing this class, more or less online. Wow. Plunge the room into darkness. You know what we're going to do? We're just going to watch movies for the rest of the semester. Yeah. I like that. It's kind of a high school thing to do, but whatever.
Dramatic pause for coffee.
Mm-hmm. And waiting for the machine to come up. No, because I'm constantly looking at like, how am I wearing my belt? Mm-hmm. God. How's this shirt look? It's a little bit wrinkled. I tried to I didn't iron it, but I started trying to smooth it out earlier. See, if you had been in class today, these are the things that you would have enjoyed. And then I post a 49-minute video and realize, God, fully half of it is me just drinking coffee and standing around. Whatever.
That's why I started coming every Monday, Wednesday, Friday.
Because you wanted to enjoy
Yeah.
standing around drinking coffee. Yeah, no, I appreciate that. I mean, we have a number of regulars now. We could put more of the class in, but not the entire class. So, I digress. I never digress, but I digress. There was a whole story yesterday on public radio out of Springfield talking about the psychology of people trying to recover from the pandemic and getting back into larger groups and thinking about, the Molec and Cell lab. We would routinely have 16 or 17 people in there, and just what's that going to feel like when we get back to that? It'd be a little bit interesting, and we have to kind of stepwise take it because we need to get beyond this. But I further digress. Okay, this is a lovely fluorescent micrograph in which we're staining for actin, which we're about to get into, and microtubules. So here is microtubules that are making their way out, and so we can imagine vesicles going this way and that way. This is part of the trans-Golgi network, and so there you go. And intermediate filaments. Yep, poor dude. All right, then we got, all right, so there are cilia there. Some of them are made with with actin, though I know it. The centrosome and the cell dividing, these are things that microtubules are doing. And here is the microtubule structure. So we've got the alpha and beta heterodimer that makes up the subunit. And you have the plus end, that's the end that you build up on, and the minus end is somewhat more static. And you form these little tubes. So it's very much like a Twizzler. Yeah. The microtubules. Yeah, we get it. All right, so there are all sorts of things that microtubules are able to do. But regardless, and some of them go, whatever. Microtubules will just autocatalytically begin to bind up. They do have GTP they bind to, and it helps them do a little bit faster, but it's not critical, it's not necessary for it. So here we have the drugs that affect microtubules: Taxol, colchicine, vincristine, vinblastine. There we go. So we got the ability to inhibit disassembly or inhibit the assembly. And all right. So one of the things that we've been going here with the motor proteins. A key place to look at motor proteins has been in neurons, because that axon is a pretty long cell. And if you think about this, and sometimes there's things that say it probably ain't good to think about too hard, because you're going, man, you have vesicles filled with neurotransmitter. They're being built back here where the Golgi is, and then they got to make it all the way over to here to the axon terminal. And I got some long-ass axons, and so do you. And think about a giraffe. That is a really long axon. And those vesicles have to march their way down. And so I got some that started last week, and they're making their way to the end of the axon terminal. And a question people have actually asked, you can have a minus end directed motor heading out that way, and then you got a vesicle coming back in, or the minus end's heading in, and you have a vesicle that's heading out, that's plus end directed. If they're on the same microtubule, what happens when they meet? And there are a number of ways you can lay out microtubules on a glass slide and affix little beads to kinesin and dynein and basically set up a train wreck. And what you see happening is here are a couple of guys that are moving, and there they go. They move pretty slowly, but they do move. If you set them up where they're coming at each other, and you're sitting there watching under the scope, eating your popcorn, going, it's going to be a wreck. They actually rotate around the microtubule and slide right by each other. So we definitely have trafficking going on and movement back and forth. So here we are. I hope I didn't screw it up. And I did. No, I didn't. So dynein is the minus end. Kinesin is the plus end. And they have the ability to bind to the microtubule, and then they have an ATP hydrolysis system. And here you can see the little dynein wall. See? Kind of like a little, I don't know what. All right, so we have all kinds of cargo moving back and forth along your microtubule lattice. What we're trying to show here, don't know, don't care, fine. And this is a game of cellular pickup sticks. Actually, this is some of the three-dimensional models of the kinesin heads as they're making their way along. And so here are some cilia. Everybody wave, you can do the wave. And that is a microtubule. And so, just to show, we have the power stroke, and perhaps this is why I don't get into it. It's a fairly complicated thing when you do a cross-section of a cilia or a flagella. You see a whole set of microtubules, and this is actually where the microtubules, rather than forming a single tube, they form this kind of a tube with a little comma around it. And they then have a whole set of dynein motor proteins around them. And they do this. So one of the ways this was determined is if you take microtubules and you have them isolated, you have this sliding back and forth. But with a connecting protein between the two microtubules, as they slide, one end is anchored, one end is moving, the whole thing will bend. And then you can bend back and forth. And so this allows for the whipping action back and forth of the flagella. And that's all we had to say, thankfully, about microtubules. We're now getting into nerd rope, I think, is my best example for this. That is the actin microfilaments. And believe it or not, we're going to go back to the board. But we do have a red, white, and blue actin microfilaments. Notice there is a minus and plus end. They're not nearly as dramatic in terms of their assembly as microtubules. And so I stand by my microtubule is a Twizzler analogy. I don't have a great microfilament analogy, but nerd rope kind of makes me think that. Do we know nerd rope? Do we know nerds? It's a little balls. And rather than forming a tube, it is, in fact, a set of actin molecules that are located up. Actin looks a little bit like a jelly bean, or actually like a kidney bean. And you start stacking them up. And so these are microfilaments. Now, the microfilament story, rather than focusing on spindle fibers, which are strictly microtubules, microfilaments, we can look at a number of examples. One of the great places to look at microfilaments is in muscle cells. That might be a good thumbnail. Probably should have better editing software, but then the entire class would be about 15 minutes. I mean, the entire semester. All right. Now on to our third class, microfilaments. These are made up of a protein known as actin. Now, if you recall, in our differential extraction experiment in the laboratory, which we don't have videos of that so much, sorry, for those of you that subscribe. But we were extracting proteins, and we talked about the fact that when you pop open the cytoplasm, there are some major proteins in the cytoplasm that have very diagnostic, real obvious molecular weights. Actin and tubulin are biggies. And with muscle cells, man, they are packed with actin. And so you can tell a mile away that, oh, you have popped open the cell. Whether you can tell it's a muscle cell or not, quite come close to it. So actin microfilaments. It is a single filament, so we don't have that protofilament business. And it is, again, an autocatalytic assembly. There is a plus end and a minus end. The plus end is the more dynamic end. The minus end is the more static end. They are single filaments. And some of the things that you would know actin microfilaments from. Remember after cell division, or after what we think of as karyokinesis, the division of the chromosomes, we have cytokinesis. And one of the things that is observed is the formation of the contractile ring. So you got a ring that is pinching off the two cells after the chromosomes have separated out. This contractile ring, this is actin. Actin is not so much used as a vesicular transport system, but I'm sure when we start looking at the figures, we're going to find a massive figure that says actin is critically used for vesicular transport. But I don't think it is, so there you go. There is one major motor protein for actin, and that motor protein is known as myosin. That is the major ATP-dependent motor protein for actin. And as we're about to see, when we start looking at myosins, you can break them down into two broad categories. We've got myosin that is specific to muscle. And it turns out that myosins, actually, there are multiple genes for it, but there is a muscle-specific myosin. And then, you probably never guess what the other one's called. Non-muscle myosin. So, the muscle myosin, we'll be getting into this when we start talking about muscle contraction here, probably on Friday. But non-muscle myosin is found pretty much in every single cell. Non-muscle myosin is responsible for the ability of actin microfilaments to carry out different slidings and movements. And so we got the contractile ring. We also have various kinds of amoeboid. I have no idea how to spell this. Maybe amoeboid movement. And one of the things I think is interesting to think about is the fact that there are a whole category of actin binding proteins with some interesting names that give rise to some of the ways actin is operating. So actin forms this big network in the cytoplasm. It can pull on things. It can actually push out using the myosin, kind of a similar kind of model here where you have myosin attached on one side and then the contractile ring is pulling in on it, various things like this. But there are a whole range of actin binding proteins. And they can be capping proteins that will inhibit further elongation. There are severing proteins. There are cross-linking actin binding proteins. And one of my favorites, and I think it's a really evocative name, is an actin binding protein that is known as gelsolin. What this does is interact with actin microfilament structures, and depending on its state, it can cause the cytoplasm to become quite jelly-like, boing, 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 or a good bit stiffer. All right. So what this means is that you can have parts of the cell that are actually quite squishy and other parts of the cell that are starting to firm up like a really serious piece of jello. So imagine you've got some kind of amoeboid-like movement. You can have the back end get pretty stiff the front end can be pretty squishy and move out. The back end then is going to become squishy and pull up, and then the front end can move out. And so this is the way you can actually have the cytoplasm change its texture and structure and affect the cell shape change, which is a really big thing. So actin microfilaments are doing a whole bunch of really important kinds of structural changes like that. Let's see. Yeah, just for the heck of it, in the last few moments, we'll look at a couple of the figures and then finish up with muscles on Friday. So actin is really important, particularly for cell shape change. And you may not think too much about cell shape change. And I have to say that as a fully functioning adult, there are phagocytes that are able to move through blood vessels, and they are doing amoeboid type stuff. But, if you were to say to me, okay, you're going to have to halt all amoeboid motion today, at least initially I'm going, okay. We don't have, as a fully functioning organism and fully grown, a whole lot of cell shape change going on. I mean, there's some, but probably not a whole lot. We've got cell movements and stuff, and we have contractions. But I'm talking about the actual shape change of the cell. But during embryogenesis, this is a major, major deal. The ability to cause infoldings and all these different shape changes happens because the cells change shape, and because they are attached to each other, that shape change results in an entire epithelial sheet being pinched in to form like the notochord. So cell shape change is a really cool phenomenon. All right, we have again the dynamic instability, the plus end, and there is a GTP associated with it, and it does hydrolyze, but we're looking at tubulin again. What the heck? Oh, there we are. Wait. Back up. Where are we? All right. ATP, DFA. Oh, wait, looking at the comparison. Since some of these are some of the different models of the actin microfilaments doing treadmilling versus dynamic instability. That's what I'm showing. All right, we'll actually pick up on this on Friday because there's a couple of interesting things. There are some drugs that affect actin filaments. Phalloidin, which is actually a mushroom toxin, might be interested in that, from something known as a white death cap, Amanita phalloides. Don't eat that. Cytoclazin is preventing polymerization. Latrunculin, which I think is a great word, but I've never actually used the drug in any experiments, is also affecting the blocking polymerization. All right, here is this biz about some of the different kinds of proteins that are involved here. We have cross-linking proteins, side-binding proteins, capping proteins. There's myosin, bundling proteins. There are things that are going to sequester the individual monomers and keep them from forming up. And there's an old-fashioned term you may see in older books and journals and things, what is referred to as F-actin and G-actin. And I think it's because they didn't know what the hell they were talking about. Regardless, F-actin is what you refer to as actin that is in a filamentous form, and G-actin is the form where it is not polymerized in these long structures. It is globular actin. And there's all kinds of terms in the actin biz. So we have myosin motor sliding and lamellipodia and filopodia, all kinds of ways of pushing out. And then here is the little walking feet of myosin. And that actually leads us up to the muscle. There, which is where we're going to pick up on Friday and talk about muscle contraction. All right, so we're making our way through. I apologize that if I was somewhat trivial and talking with the audience here. maybe low blood sugar. So what I'm worried about is you can start to statistically analyze my shirt and how many times I wear the same shirt.